Alright, so then Protista are going to be the kind of the baseline of all the eukaryotes. So they're the ones that are going to allow for plants, animals, and fungi to start to evolve. We're going to talk about Protista or protists on Monday. But what you need to kind of see is that there are three main types of protists. We have plant-like protists, and they're eventually going to evolve to the plant side of the eukaryotes. We have animal-like protists, and they're going to evolve eventually into true animals. And then we had, guess what? Thank you, fungi-like protists, and they evolve into the fungi side of things. Okay, so the protists are really a catch-all kingdom. They're very diverse because there's three different kind of varieties of them, whereas bacteria and archaebacteria, yeah, they're diverse, but there's not like three really main types. We're going to talk about a lot of characteristics of bacteria and viruses today, though. Notice, do you see viruses on the tree here?
No.
No, because are they living?
No.
No. Okay, they're definitely not living. They have some abilities to reproduce, but it's not by themselves. They kind of infest something else and then make them reproduce for them. So we're going to talk about that in a second, too. All right, so you're going to watch a video. This is what I want you to do. The papers that are in front of you, the colorful papers, and you have white papers. For this whole unit, we have kingdoms we're going to go through in very good detail, and then the human body. This is a huge unit. We are doing this until December the 18th, okay? So it's like almost a 2-week unit, okay? So instead of making you turn in papers every day and labs every day, it's pretty large, and I'm scared that you guys are you're going to start to lose your papers since it's so large. So you're going to make a kingdom book. That kingdom book is going to include a lot of information that we do every day. And to reward you for working hard on this the whole time and keeping up with it is a test grade, okay? It's pretty easy. Keep up with your stuff. Take good notes. Write down your data from labs and drawings from labs and anything like that in your little book, and you will be fine, okay? It's an easy test grade. If you prefer to write it on notebook paper, you can put your notebook paper, like fold it into like a little book, and you put it in that book that you're going to make, okay? So today is going to be bacteria and viruses. What I would do is I would do something like this, and you can keep this every day. I can give you more white paper as you need it, and you can add. So I'm just going to fold this in half. This will be the beginnings in my book, and if I have to add paper, I'll add paper later. The reason I gave you 3 pieces of paper is because there's 6 kingdoms, and so when you fold them in half, you might have 6 pages. We're going to probably need more, I guarantee it, but I'm going to start you off kind of limited right now, just in case we lose them. Yeah. If you want a large book, yeah, but I'll have to give it to you a little bit, okay? So this pretty much is going to be your title page. If you want to make it like a full book like this, you'll have a colorful sheet of paper. That'll be the outside. That'll be your title page. Yeah, and I have different colors around. You can do that. Like somebody does. You want pink? You want pink? Red? You want brown? There's yellow behind you. I already folded it. All right, go ahead. Oh yeah, I can't say what's name. That's why I didn't say it. I was gonna fold it. Okay, does everybody have a little book kind of initially started? Okay, your first page is gonna be bacteria. Go ahead and label the top of your page bacteria. If you have colorful writing utensils, use them. If not, pencil, pen is fine. Does not matter to me. So this is going to be called the Kingdoms of Life book. So as you go through this unit, you're going to add everything to this. That is notes, drawings, and whenever we get to like drawings of organisms and labeling the features and characteristics and anatomy of them, and even labs or worksheet stuff, okay? So this is going to be like an all-inclusive little book of everything kingdoms related, okay? So if you want to design your cover later, that is fine. The cover should be probably interesting, but you might just remember you want to call it the Kingdoms of Life book by Jermaine, by Alex, by Emily, by whoever, okay? This is by you. You're making this book. You're expected to keep up with it because at the end of the unit, you are turning it in for a test grade, okay? Yes? First page is going to be bacteria. Kingdom, eubacteria, but bacteria is what we're talking about, okay? All right, so we're going to watch a video. On the first page, you have to write down 15 different points from the video. It might just be little details. It might be things you learn. whatever. Say the first video you're watching is on bacteria. You have 15 bullet points you're going to make on the first page of your book. Got it? We're rolling. We're rolling. Okay. First video is bacteria. Here we go. Do I have the volume up? Huh?
So over our winter break, we did a little skiing, and we also got a little sick. Petunia got strep throat, and Pinky had to get a root canal. So what do both of these have in common? Well, they've given us some inspiration, but besides not being fun, both of these, they were caused by bacteria. And just a disclaimer, an amoeba does not have teeth, and they do not have a throat either. But we really did get sick. Bacteria can make most people shudder. When you think of germs, and why wouldn't we? Because many types of bacteria, they can make you sick. Strep throat, it's caused by this little bacterium called Streptococcus. And it's no little bug because it can make you very sick. It produces toxins that your body has to fight off. In addition to causing mayhem as it grows in your body, many types of bacteria can do this. Oh, and for Pinky's root canal, the root canal was needed due to a cavity that got out of control, and bacteria plays a major role in tooth decay. We both had to get antibiotics. Your immune system, it can fight off many bacterial infections, but sometimes it needs the help of antibiotics. And antibiotics are effective against many types of bacteria. Most forms of antibiotics, they're going to target the structure of prokaryote cells. Bacteria are a great example of
Oh, we have a question. Generally, what does an antibiotic target on a bacterial cell in order to stop it from causing infection? Prokaryote what? Structurals, which is, in this case, it says plasmid, cell wall, or cytoplasm. Cell wall, thank you, good job. Definitely the cell wall, so that's a good one. Antibiotics target the cell wall. We're awesome.
Prokaryote cells. When you think of pro, think of it rhyming with no. It has no nucleus, and it has no membrane-bound organelles. Bacteria still have a cell membrane and genetic material like the other cells, but no nucleus, no membrane-bound organelles, and antibiotics will target them.
All right, so you remember that pro means no nucleus, right? All right, that might be a good factor, right? Prokaryotes, no nucleus. That's, some of these are review facts, and I'm just listening and writing a little facts that she says as I go. I'm already on number 4. I don't know about you guys.
Thing though that is interesting about antibiotics, if you've ever been on them before, you might notice that sometimes they can make you feel a little bit sick to your stomach, or they might make you feel like your appetite is negatively affected. And here's the thing, most antibiotics that are broad spectrum, they're not very super selective. They destroy the bad bacteria, but they also get the good bacteria. And yes, you did hear me right, good bacteria. You actually have tons of good bacteria in your body. In fact, you have more bacterial cells in your body than you have human cells in your body. That's a little bit scary.
Whoa, so more bacterial cells than human cells in your body. So you're mostly bacteria. That's kind of cool. Maybe. Just call me Miss Bacteria. Okay. So seriously, you have 10 times the number of bacterial cells on your body than human cells. One reason is because the good bacteria take up all your free real estate, which is like your skin, pretty much, or any surface on your body. So the good bacteria inhabit the real estate of your body, so the bad bacteria have no place to live on your body. And you want that to happen because that way the good bacteria are staying around and bad bacteria don't have a place to live, so they can't come and affect you.
Bacteria are especially found in your digestive system. They help you break down food, and they also help you synthesize certain molecules, like vitamin K, for example. And they keep other microorganisms from getting out of control. For example, you know bacteria is everywhere. You know what else is everywhere? Fungus. Yeast, for example, is a type of fungus. And yeast grows everywhere. A possible side effect of taking antibiotics is that you can develop a yeast infection. Why? Well, if you kill off the bacteria, you're taking out the yeast competition. The yeast doesn't have to worry about the bacteria anymore, and it can grow out of control. Have you ever?
All right, so bacteria serve to Never finished that question. It's got to be that one, right?
I wondered why the people on yogurt commercials look so happy. Well, some foods, like yogurt, they need bacteria to help produce them. And the thought is that by consuming some of these foods, you might help contribute to this bacterial flora that lives in your digestive system. And we can only wonder if maybe that's why they look so happy. Bacteria are also one of the most important decomposers in food webs. Scientists do a lot of genetic research using bacteria.
All right, so they didn't really say it, but you know yogurt has something called probiotics. You've heard of this, yes? Pro means to support, and biotics means life, or bio means life. So you're supporting bacterial life, and that actually really is healthy for your digestive system. That's why people eat yogurt a lot. So kind of after an antibiotic, it kind of wipes them clean of their bacteria, so they eat probiotics to get them regular and get them feeling good again. Because bacteria really do a lot for you in terms of digestion and like making vitamin K. That was a really good example that she used. So now they said that bacteria decomposers in a food chain. That's another good fact. Decomposers.
Bacteria as well. Bacteria are cheap, they're quick to grow in the lab. In fact, some exciting research to look up on the web is to see how bacteria can be used to clean up oil spills. Bacteria already can do this, but the engineering part comes in when you're trying to figure out an effective way to have bacteria clean up these oil spills without them harming the environment at the same time.
Okay, well, sorry, one second. So that's a really cool fact. Bacteria, so you guys remember the BP oil spill, right? Yeah. Okay, there's actually bacteria that actually exists in the water, and the bacteria are, oh gosh, what's it called? They're oil eaters. There's something trophs. Oh gosh, I've forgotten what they're called. They're not called oiltrophs, but it's something similar to that, so I'll go with it. So they're oiltrophs because they eat oil, and when they eat, they eat the hydrocarbons, and when they eat it, the oil goes away, right? And that's really good. Sometimes they excrete other toxins, or they poop carbon dioxide or methane and stuff like that. So we try to let the bacteria eat the oil, but sometimes they release bad gases. So they're actually biogenetically engineering, so like making GMO, genetically modified bacteria, that eat the oil, but just pretty much poop out water. Like, can't have too much water, right? So that's what they're trying to do. So they're trying to biologically engineer these bacteria so we can use them for things like that. Okay, so bacteria can, check all, okay? Can they help with digestion? Yes. Yes. Can they cause you illness? Yes. Yes. Can they compete with bad bacteria and fungi to protect your body? Yes. Yes. Can they be used to eat away oil at an oil spill? Yeah. Yes. Yeah. Synthesize vitamins for your body? Yeah. Yes. They can do all those things. Those are some fun facts for bacteria. Yes.
Something to think about the next time you want to call bacteria the bad little microbe. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
I got 11, but I could have rated more. All right, let's write down some more then. Okay, let's write down some more from, let me get here. All right, as we go through, you're going to write down a couple more, and I'll tell you, I think important things from the notes, and then we'll jump on viruses. Okay, you already know they're prokaryotes, right? Yep. You already know they have a nucleus or no nucleus? No nucleus. No nucleus, right. Do they have ribosomes? Yes. Yes, fun fact, write that down. They have ribosomes. Why in the world would they have ribosomes? What is the job of a ribosome? To make what? Yes, synthesize proteins. And that's going to be, just an organelle-based question, but yes, they definitely need themselves to make proteins. And I'm going to scroll up a little bit, and you guys can draw yourself a bacteria on the little, right underneath maybe on the next page or on the same page if you have room, it depends. If you need some more space for your 15 facts, I'll go to the next page. Draw yourself a bacteria. Use your artistic skills. And I'll help you with the labeling because it's kind of hard to see. So I'll help you with the labeling. Because they would totally love seeing your books. The anatomy class did that. They made little cartoon books on the heart and like how the heart works and the circulatory system works, and they were awesome. And the little like third graders were amazed. So if you guys do a good job with it and you want me to share it with them, I totally will. Yeah. I'm gonna help you with labeling. Yeah, that's the hard part to see. I'm going to erase my notes over here and draw over here. Alright, so you've got your kind of circular shape. I'm going to tell you how many rings you should make around it, okay? There should be the first ring around it. Alright, so the inner ring is going to be the cell membrane, also known as the plasma membrane. The second ring I just drew is the cell wall. There's a third ring, which is a third layer. Third layer is called the capsule. Alright, so you guys should be drawing these because this is going to be in your book, okay? So we've gotten the capsule, the cell wall, and the cell membrane taken care of. What's on the inside? Alright, so I got little dots, right? Those dots are the ribosomes. What else do I have on the inside? Cytoplasm, so the wavy little waves. Cytoplasm. What else is on the inside? Okay, ignore the granule, ignore that one. The DNA bundle is kind of weird. You can just call it DNA. There's actually some kind of crazy curly stuff in here. That's our DNA, and they just call it DNA bundle because it's all bundled together. Alright, so I'm going to draw a little wavy, curly, cute looking stuff, and that's going to be our DNA. In addition, though, bacteria usually have that circular piece of DNA, which was called the plasmid. Good. I'm just going to draw a little circular piece of DNA out to the side because that's something special that contains some different characteristics, and they usually like to share their plasmid with other bacteria because it helps them survive. Okay? So this little circular thing was called the plasmid. And then what am I missing? The what? Flagella. So there's a big long tail. Flagella. Is that everything? Oh, the pili. I'm sorry. I see what you're saying. Okay, so bacteria have a flagella. What do you think that flagella is going to help them do? Move. Okay, that's pretty easy. They also have something called pili. So they have like little tiny hairs that are kind of exist on their outside. So there's little hairs all the way around it, like stubble. Okay? Those little hairs are called pili, and they help them stick to other surfaces. Like, for example, it helps them stick to the inside of your nose or your throat or your stomach or to your skin. It helps them stick. All right, that's all I'm going to label on our bacteria here. So now we got all the important stuff.
What is that?
The granule, that one is actually like, it's like a little storage molecule of like carbohydrate. And sometimes we talk about that, but they have like little food stores on the inside. But I'm not going to ask you guys about it on your test or on your PSC or anything, so you're good. Yes, question? They have, in this case, they have, what's it called? They have 2 flagella, but it's called something special. But sometimes they'll have 2 flagella, sometimes they'll have 3, sometimes they'll have 1. All right, so my picture's on the board. If you can see it there, you guys have your stuff labeled, yes? Mostly, it's on the board. You got this. Ignore the granule. Yeah, it's a. Oh, this is our DNA. It's a little bundle of chromatin, which is not in a double helix form. It's just kind of floating around. Okay, now bacteria. These are some more facts. Some of these facts that you probably added from the video. The red stuff is the important stuff. Okay. So did you guys say they cause disease and decay? All right, so you said like maybe streptococcus, that was strep throat they were speaking of. So they had disease. Decay, what's another word that would mean decay, like to break down? Right, so that's what they said on the video, and if you have it, you're in good shape. So they're good at decomposing things, and that's really good because if not, what would happen to all the nutrients in like dead bodies? They'd just be chilling there. They would never get back into the earth. We need that nitrogen cycle. We need that carbon cycle. We need the oxygen cycle. We need the water cycle. We need every phosphorus cycle. We need everything to recycle. If not, we can't get any nutrients ever. All right. The only other one on this one that you've not written down is binary fission. That word should be familiar. This should be a fact that you add to your book. They reproduce by binary fission. And I have a picture of it happening down here in just a second. Binary fission, what do you guys think? Is that asexual or sexual reproduction?
Asexual.
Right, definitely asexual. So you need to know this is asexual reproduction. Bacteria, their cell grows, they chop it in half. Their cell grows, they chop it in half. And so we already talked about their cell wall. Their cell wall is actually usually attacked by the antibiotics. So their cell wall, though, generally is strong. So antibiotics have to be strong in their ability to attack it. So the reason that it's strong is because bacteria, they're floating around all kinds of places. They don't want their cell wall to just burst because they would die. So that's why they have to have a pretty thick cell wall. This is typically what bacteria looks like, just like ours, like almost identical, right? Yeah. And then you can see this bacteria here, and then how it's split in half through binary fission. So binary fission is definitely a way bacteria reproduce. And I'm going to show you this little reproduction video. If I find it, this one. All right, it's kind of small, so I have to. What? Really? Why won't you let me see it? Well, you guys will be all right. That's not hit the lights.
Go to the control buttons on like the top right.
Yeah, it's not gonna be pretty though. See? It doesn't do it. That's not helpful. It's not helpful. Okay, let me see if I have another one that's better. Oh. Okay, so you can see the bacteria. They're multiplying. How are they reproducing right now? Asexually. Okay, let's watch again. Would you say that is meeting their carrying capacity or they're exponentially growing? Right, definitely exponential growth. Is that an S-shaped curve or a J-shaped curve? J. Okay, it is definitely a J-shaped curve, okay? Draw yourself a J-shaped curve next to asexual reproduction, wherever you wrote it down.
Is that in real time?
That is, no, that's actually sped up some, but they can reproduce every, oh gosh. They can reproduce it's much more quickly than anything else. It's like every 30 minutes they can double, and so like in a day they'll get up to like thousands and thousands of cells. So it's because as one reproduces and it makes another one, it can, you know, those that are made can then make another one as well. Okay, so I told you there's not much to write here. The second type of reproduction that some bacteria can do, not all of them, is conjugation. You're familiar with this, yes? This is a term we've spoken of before. Write down the red terms are what I'm kind of highlighting here. Those are the words I need you to write, and then if you want to write anything else, you of course can. Conjugation is really cool. Okay, so bacteria, we already told you, what are these little hair-like things around? Pili, right. Okay, so pili are just like little hairs. Well, there's a special pili that some bacteria have. Not all of them have it. It's just some of them, and it's called a sex pilus. So they shoot it out at another bacteria, and they say, "Come here, baby, I need you." And then they force that other bacteria to take its DNA, so they can send through their pilus their little circular. What's that circular DNA called? Plasmid, right? And actually, usually that other bacteria. With me? Usually the other bacteria wants that bacterial's DNA because that plasmid usually contains a gene for antibiotic resistance. So, like, why would that other bacteria not want it? They're going to live forever until the antibiotics change, of course. But that is a special type of bacterial reproduction called conjugation, and that one is definitely sexual because you can see it's called a sex pilus that helps it do that. Mm-hmm. So that's a special way that bacteria force the exchanging of DNA. All right, so the asexual type was called what? Binary fission. Sexual type is called conjugation. I'll give you guys another second there. All All right. When I see most of us are okay, I'm moving. These people took my little clicker yesterday and they didn't bring the other thing back. Okay. Okay, do not write down these right here, but I'm going to talk about them on the next slide, okay? So bacteria are classified in 3 different characteristics: the way they are shaped, the way they eat, and something called a gram stain. So let's look at their shapes. These are the 3 shapes of bacteria. The red stuff is the important stuff. Write down, you can just put a title like characteristics of bacteria. Yes, so characteristics by shape, and then the 3 types of shapes. And you can even draw the little shapes if you want to. Alright, so the shape coccus is a round shape, and you can kind of see that illustrated like here. It's a spherical shape. Rod-shaped is bacillus, and I'm going to tell you that you have to know what these shapes are. And you have to know kind of what they are related to. So the coccus shape is the round shape, and you look at all these letters. Round, 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 round. The C's and the O's are round shaped, so that's how I remember that coccus means round. If you need to write down that tip for yourself, write it down because that's a memorization kind of thing. Or just think coccus circle, CNC. Bacillus is rod-shaped. Which of these letters are rod-shaped?
L.
L's, thank you. The L's are long, like a rod. Here, this is kind of hard to see, but it's just a long rod. That's it. So that's just kind of elongated shape like that. Spirillum, well, that kind of just sounds like the word spiral, so that's pretty easy. Could you just say, oh, that's just a spiral? Okay, so it kind of has a little spiral-looking shape to it. Thank you. So spirillum has a spiral shape to it, like curly hair. Thumbs up when you're done here. I'll do this on my stripe. Here. We're making a book. Alrighty. Okay, now an example of an example of coccus is the bacteria called Streptococcus. What did we say that caused? Strep throat. Oh, it's right there. Sorry. Okay, so that causes strep throat. Staphylococcus. Well, what shape is it based on the name? The circular round one, right? So it's still a circular shape. It's just Streptococcus causes strep throat, and then Staphylococcus causes staph infections. So all the disease names that we say, they're actually related to the scientific name of the bacteria. So that's why, it's relevant for us to talk about that now because we know what classification is. So what I would write down is an example, Streptococcus or Staphylococcus. One of them will do. And that causes the disease, the disease. Strep throat is not a disease. The illness, strep throat or a staph infection. And then they just have different arrangements. Sometimes they arrange themselves like beads on a chain. Sometimes they clump together in little clusters. So there's just different varieties of how they appear. So like I said, I'm just trying to shorten your notes in terms of writing down the relevant things I ask you to write. Thumbs up if you're good here. All right, I'll give you guys another 5 seconds. Okay, now we just did the characteristic shape. Now we're doing the second characteristic, mode of nutrition. So the second characteristic, mode of nutrition, write down the 2 types of modes of nutrition they can have. And then I'm going to give you a detail that you want to include for each of these. Oh, did I skip? Oh, yeah. I don't have really good example pictures of the spirulina.
This pocket
No, this is so we just did the 3 ways they classify. So we just talked about shape. They classify Coccus, Bacillus, or Spirillum. Now we're going to go over the modes of nutrition, and then we're going to go over the Gram stain test. So now we're going to talk about characteristic mode of nutrition. Heterotrophic and autotrophic. These are familiar to you. Heterotrophic means that they have to consume. So an example for this one would be oil-eating microbes. Those little bacteria that we talked about previously from the video. So we're done heterotrophic and oil-eating. And an autotrophic already has an example. Cyanobacteria. What does the prefix cyano mean? No, that would be awesome, but no. It actually is a color, right? Cyano means blue, green. It's kind of like an aqua. So this guy right here, first of all, can you see his little shapes? They're not rod, they're built together. They're actually just a long chain of circles. So what's his shape? Coccus, right? He's a coccus shape. He's blue-green because that's what the cyano refers to, and he's photosynthetic because he's green. So what do you think? What kind of pigments could be on the inside? Chlorophyll, good. There should be some sort of chlorophyll on the inside of the cells, which makes it have the ability to do photosynthesis. Now, it is not it's not going to have like a huge amount of chloroplasts because that's an organelle, and prokaryotes don't have organelles, but they have a little slightly different way they can do photosynthesis. Yes?
Do we have bacteria in our body that do photosynthesis?
Probably not on our body, but like any animals that live in areas where it's really kind of moist, they have bacteria that live on their fur, and and it's like sloths and not koalas, but I think it's sloths. They have a symbiotic relationship with some bacteria that live on their fur. And these are just some pictures. Down here are just some pictures. You got oil-eating microbes that eat the oil, and then nom nom nom. You can see that they're eating you can kind of see they're eating the oil. And then this is an example of a beaker, it looks like, with oil in it, and then 7 days later, the oil has kind of disappeared, and that's because the bacteria were eating it. And they do a very, very good job of oil, and that's why they use them in bioremediation of oil spills. All right, last one is the characteristic is called Gram stain testing. And there's only 2 things you're going to write about Gram stain testing. Our Gram stain test. If you have time to. Yep, I told everybody you weren't here. I'm going through the details very minimally because we have to do a lot of content about a little bit about everything. So sometimes my notes are just more detailed than they need to be. Yeah. So Gram stain test, the red words up there. You can either have a Gram stain test that is positive or negative. All right, so when you look up here, you see, okay, the Gram stain test is testing for the stuff in the cell wall of a bacteria. The stuff in the cell wall of a bacteria is called, get ready, peptidoglycan. Peptidoglycan, okay? So if it's positive, what do you think it's seeing? What do you think that Gram test is saying yes to? It has peptidoglycan. Peptidoglycan. I always murder that word. It has the peptigo stuff in the cell wall. If it's negative, no. Does not have it in the cell wall. Yes?
What is the word peptide?
It refers to like amino acids mainly, but not in this case.
So the medicine Pepto-Bismol, does that have that in it?
No, not that in it. Pepto-Bismol has bismuth in it. It's an element that helps with, it's a metal. So Bi on the periodic table is the main element in Pepto-Bismol. One of my students last year, maybe Cam, wanted to synthesize bismuth from Pepto-Bismol, but he would need like 1,000 bottles of Pepto-Bismol to get like a gram of bismuth. It's so hard to get. So we were gonna do that. I was like, I don't know. It's gonna cost too much, Pepto-Bismol. Okay, I think I've got like a break for you guys in just a second. Okay, last thing. This is the last thing for bacteria. They are so important, okay, not only to us, but to the world and to helping with nitrogen cycles and ecology. So you need to know these 4 categories of their importance.
This was all supposed to be on one page.
Oh, no, if it's on more than 1 page, that's totally fine. We're making a book, and if you can have about 1 to 2 pages per kingdom, that's fine. If it's more than that, I don't care. I'll give you more paper. Oh, and your book is going to be turned at the end of this unit, which is on December the 18th, and it's a test grade if you do a good job. So I'm trying to reward you guys for being attentive. I know it's Christmas-ish time, and some people are excited. Santa never comes to my house anymore. All right, so you got your 4 main categories. Thanks. All right, so food production. I'll give you an example of this. Write down your examples. Here we go. Food production, cheese, yogurt. Anything else you guys can think bacteria are in? Milk, yeah, cultured milk. Ice cream, so any dairy products. I just write down dairy, that'd be easier. Digestion in mammals, that's just going to help us break down our food. Break down food, which is like, duh, I know what digestion is, though. Nitrogen fixation, put a star by that one. If you don't know anything when you leave me, you best know that nitrogen fixation is done by not us. Who does it? Nitrogen fixing bacteria, right, bacteria do it. And that allows for nitrogen in the air to go into nitrogen in the soil so that the bacteria can take it up. Okay, so this is going to allow for air nitrogen to go to the soil nitrogen so that plants can take it in. And then who eats the plants? Us, and then that's how we get nitrogen in our bodies for all those biomolecules that contain nitrogen. If not, we would never be able to get nitrogen and we would all die. So thank you, bacteria. We need you to do this for us. Decomposition is the last one, and this is just going to help with recycling. Is that spelled right?
Yeah.
Yeah?
Yeah.
Okay. Recycling nutrients. What are those 5 main nutrients that we need in our bodies? Carbon, hydrogen, oxygen, nitrogen, phosphorus. Good job. So we definitely need all of those, and so if bacteria don't recycle it for us, then we don't get them, and then we die. So thank you, bacteria, again, for recycling the matter and the elements for us. You get a stretch break in just a second, okay? Okay. Not write this down? Oh, did you guys previously write down the antibiotics thing, right? Antibiotics are used to treat bacteria. These are just a couple of those examples of, this is tonsillitis, where they've got inflammation of probably strep throat. This is bacteria that's causing, who's ever heard of, oh gosh, what's her name? It's not Salmonella Mary. No, I can't remember. I think it's like Typhy Mary or something like that. She was a cook back in the 1800s, and anybody who ate her food got salmonella. Okay, got typhy. Okay, so what they were trying to figure out was where was it coming from. Well, then they finally tracked it back to her. She would travel across the country because she was a famous chef. But she had naturally, like, her aprons were dirty, and so she would just have this bacteria on her, and it would get in her food. And salmonella is something that causes massive diarrhea. Okay, people were not happy with this, but they finally tracked it back to her, which was a weird occurrence. So that was just a side note. Yeah. No, of course not. It was sanitation back in the 1800s versus sanitation in the 21st century. Very different. So now, of course, you go in the restroom and you'll see employees must wash their hands. You have to go through all that training and stuff. So that's definitely, it's all her fault. But that's a good thing. Thank you, Mary, for deciding that we need this. All right, you have about a minute to get up, take a break. A minute to get up and take a break. Yeah, she was okay. Alright guys, while you're taking a break, I'm gonna talk to you. Ready?
I'm ready.
I can keep, wait. If you guys can explain, I got all kinds of stuff to do the rest of the year. I was getting ready to say, instead of doing more notes right now, I was gonna do a lab. Are you still complaining? No, okay. So, let me tell you what I need you to do. And I got it up here. This is one of my favorite labs because you get to choose something for you that you're interested in and then try to see how it works out in terms of bacterial growth and making a new colony. So make sure you're listening for me because if you do it wrong, it's going to mess up the auger and then you won't get good results, okay? So what we're going to do is we're going to push our lab. We're going to do our lab right now. Boys in the back, please be quiet. Separate or something if you can. You're going to do your lab right now. You're going to test for the ubiquity of bacteria. Where in this school, near my classroom, is there bacteria, okay? So, yeah, don't tell me because I want you guys to write it down, okay? So you're going to test for it though. So what you have to do is you have to give bacteria a little bit of nutrients so that they can eat. And as they eat, they grow and grow and grow and replicate through asexual reproduction. So here's how we're going to do it. Each person's actually going to get their own petri dish, and you're going to decide 2 locations to test for bacteria. And on your bottom of your petri dish, you need to label that. So I'm going to show you how to do it, and then we'll set you free, and you come back. We'll finish up here. So let me show you this first with my marker. Okay. So we have little baby petri dishes all for you, and I prepped these about 5 days ago, and some of these have mold spores that have already contaminated them. So the bacterial fight the mold, and somebody will win. We'll figure out who it is. Okay. So what you're going to do is you're going to so your petri dish does come apart, right? You don't want to pull it apart because the bacteria and the mold spores in the air will contaminate it. Okay. So you only want to keep it open just long enough to put on a sample that you need. Okay. So on the bottom of the petri dish where the auger is, on the outside, you're going to take a marker and you're going to draw a straight line. Side 1, side 2, okay? So area 1 and area 2. It doesn't have to be huge letters. It's just for you to know what is what. And on the bottom, someplace underneath 1 and 2, put your initials so that you know that this is your plate because they're all going to look the same at this time, okay? So you're going to take a sterile, meaning clean, cotton swab. It's just literally a Q-tip, okay? If you want to test like the surface of the desk, well, that's dry. So you're going to take the Q-tip and you're going to put a little bit of water on it, wipe it on the desk, and then you're going to really, really, really lightly open up your petri dish and just take the Q-tip and wipe it on the auger. Don't dig it in the auger. It's like Jello, okay? It's gonna crack. So just wipe it on there, twirl it around, wipe it. That Q-tip has another side on it, okay? You're going to go find another place. Now, let's say you go to the bathroom where there's like a wet sink. That's already wet, correct? So you can just take the Q-tip and wipe it on the wet sink, and then go to your side 2, wipe it on the side 2 and twirl it on side 2, and then now your Q-tip can go in the trash, okay? So then after this is done, you're going to just put it back on the lab station with make sure your name's on it, and then you're going to answer. I have a couple questions on the next slide, and then we'll finish our notes, okay? This should take us no more than 10 minutes. I'll make sure you guys have Sharpies unless you guys have your own. So, questions?
You get stuff from outside?
I have to let somebody let you in. If you can get out, just out and right there, that area is fine. Okay, you're going to stay on this wing. This wing.
The swamp and water.
No, we're not going that far.
Right
Alright, let me grab my petri dish for you Just grab and go. Grab and go. You have 10 minutes.
Sharper she is.
You go out of my class, I'll be back in about 5 minutes. So this part right here. Alright, go. Take your time if you want to. I know what to do, but I don't know what strawberry, fuchsia, mayonnaise.
I want to swap them. Green.
I can't even see this color. What? It's not fair. No, I won't see it. I'm very so not even.
Are you sure that's right?
Is he giving you a Sharpie?
I want that
Are
red
there
one.
more Sharpies over here?
I want that red one.
That's my bad.
I am not
How much?
You have to draw it on both, right? Oh, there's only one. Never mind. It's you.
You guys need a Sharpie. Sharpie.
So where do we? Right here.
Got him.
Asian persuasion.
Like Asian.
Right here, right?
In the dirt. Were you listening?
Oh yeah, the keyboards are disgusting. I wonder how bad the sinks are. Do the eyewash station? The trash can?
Huh?
Yeah, I'm just trying to A1, that big one.
Yeah, well, ears are, I'm sorry, your ears are at body temperature. These are going to be sitting at room temperature. You can see if that bacteria is at room temperature.
Okay.
It's full of bacteria.
I know, right?
Full of it, right?
Right there. Right there.
Well, different areas. Different areas. So this area is different types of bacteria. Dude, the bottom of people's purses is really bad too. You done? You can go to that side station. It's not. What's up? What's up? If it's not dry, if it's a dry area, you're gonna wet your Q-tip with some water and then wipe it. Chandler, where'd you put yours? Where'd you put your bacteria? On the back last station, which was Chandler, where'd you live? Never mind, I'll let you do it. Wait, I don't know sure that you're writing down your areas so you remember what's what.
It's in what is a really disgusting place?
Your mouth.
Can I swallow my mouth?
Yeah, but just think about it. Your mouth is at body temperature, and that bacteria may or may not live at room temperature. But you can try. I always think that's fun.
Do I need to just like swallow my cheek?
Inside your cheek, yeah, with a clean side of your cheek.
Bacteria lab.
It's pretty easy. On the back lab station. Alright guys, make sure you're writing down your areas on your papers with your samples back there. You label your areas 1 and 2, and then put that back there. Hurry up, no, hurry up. You have 1 minute. Let's go. The whole class is waiting on you. Thanks, guys. Make sure you write down your areas in your notebook and maybe draw yourself a picture.
You don't get to keep that.
Write down your areas.
Oh, I wrote mine on the
Write it in your book too, just in case the petri dish is lost or something. I don't know. Put your initials. Yeah. Bacteria lab. It's pretty simple.
No, I'm really good on
Areas. Categories.
What makes you think I think it's not?
Tell you now. All right, you have 10 seconds to sit down. 10, 9, 8, 7, 6. Get ready. We're gonna watch another video. 5,
Are we done with bacteria or are we still?
4. We're gonna do viruses next. 3. Sit down for me.
No.
2. Jared, do you want to sit at one? Sit at that one so you can see. Okay. All right, 1. Let's go, people in the back. We're waiting on you. All right, everybody else, in your little book that you've created, I'll wait on you. In your little book you've created, you need to make sure you leave space for the analysis of this lab, okay? So you should have written in the book, bacteria lab. You probably should have drawn this petri dish. Okay, draw yourself a little petri dish in area 1. Maybe it was the water fountain. Make sure you know what label area 1 was. Area 2 may have been your mouth. Make sure you label what area 2 is. Put that in your book so that you don't forget it or lose it. And you're going to be recording data in your book, so it's kind of going to help you record it in an organized manner. Okay, so let's see, Monday, we'll check the progress, and then throughout the rest of the week, we'll kind of maybe count colonies if we possibly can, record some of their characteristics, etc. All right, so now next page is going to be viruses. Okay, next page is going to be viruses, so make sure you leave space for your bacteria lab. What's up? I'll get you. I'm videoing right now, too, so you're good. Okay, here we go. Virus, yes, sir. Yes.
Are you gonna collect the pressure points?
I will in a little while when we get to independent work. Okay? Okay. All right, viruses. I have a virus video. Let's try for 15 facts again. I think you'll get this one on this one, okay? So, viruses, 15 facts. Here we go. Where's my remote? Yep, 15 things you learn are here.
Chances are, in your life, you've had the common cold before. The cold is extremely common. You know, it causes the sneezing and the coughing and just makes you feel awful. The worst thing about a cold is that you can take medications to help with the symptoms.
All right, you should be facing forward, and you should have your pencil in hand so you can write down facts as you listen. If you can't do that, you will have to leave because you're distracting others.
But you really can't treat the actual cold. You can't really get rid of what causes it. Why is that? The reason for this is because most colds are caused by viruses. And we've talked before about antibiotics. They only work on bacteria. And viruses are definitely not bacteria. So antibiotics have no effect on viruses. In fact, viruses aren't even cells at all. You can't say a virus is a prokaryote or a eukaryote. It's not a cell. It doesn't have a cell membrane like all cells have. It doesn't have a lot of cell components. It's a non-living structure. In fact, viruses can't even reproduce on their own. So viruses, again, are not alive. It makes them very difficult to treat. But let's talk about what they actually have. What is their structure? Because they do have a structure. One thing all viruses have in common is that they have some type of genetic material. Now, this does not mean that they can reproduce on their own, but they're going to use the genetic material and force their host to reproduce themselves. This genetic material for some viruses is the form of DNA, and for other viruses, it's RNA. If the virus uses RNA, we call that virus a retrovirus. An example of a retrovirus is HIV. HIV is the virus that can lead to AIDS. We'll get into that in just a little bit. But any virus with RNA is called a retrovirus. Viruses usually also have some kind of protein coat, also known as a...
All right, so far, what have they said about the characteristics or structure of viruses? Okay, so it's not living, it's not a cell. It's, it's got RNA as the genetic material, it's a retro, so R, retro, that's pretty easy. And then I wrote down antibiotics don't work against it. So I've got 6 facts so far. Oh, good job, guys. So they you can't treat the virus, you can't treat the cold, but you can treat the symptoms is what another thing she said, because how are you supposed to kill something that's not living? You can't. Okay, so that's what we're gonna learn a little bit more now, okay? So capsid is the next characteristic or structure piece that they're noting about. Capsid's a vocab word to think of. They can protect that DNA or RNA genetic material. Another thing to mention is that viruses are very, very small, much smaller than bacteria. And you typically are going to need an electron microscope in order to see a virus. Very, very small. Viruses can also contain other structures. Some of them package special enzymes with them. Some viruses have an outer envelope. So there's different varieties on what viruses can have, but the main thing is that they have genetic material and they typically have some type of protein coat, also known as a capsid. Now, viruses are very, very specific. So you want to think of them as they're pretty picky. Usually viruses have specific types of cells that they go after. So let's talk about viral reproduction, because we did mention that they can't reproduce on their own, and they're very, very specific for the host that they're going to go infect. So the first cycle of the 2 that we're going to talk about is the lytic cycle. In the lytic cycle, the virus, remember they're very selective, attaches to a host cell, and it's going to inject either its DNA or RNA, depending on what kind of genetic material it has. All right, so this is the lytic cycle, L-Y-T-I-C. We're going to do a little worksheet on this afterwards, so this is important to know, the lytic cycle versus the lysogenic, which is our next category. So the lytic cycle, this is a bacteria. It looks like an alien. It's got like little legs on it. It goes and attaches and kind of pushes its DNA into the host cell. So this might be like this might be a blood cell from a human blood cell, or this might be like a skin cell, something like that. So they're very specific on what types of cells they can inject with.
into the cell. You would think the cell would notice this, but in many cases it does not. It takes that genetic material and it starts following the instructions, which in this case is very bad, because what it does is it makes copies of the virus. And it starts making so many copies of the virus that usually...
Alright, so the host cell has no idea what's going on. It's like, what? I got some new DNA. That's cool. I'll just do whatever you want me to. So it just keeps making the virus DNA unknowingly, and then all these virus little babies are building up inside of it. What do you think is going to happen next if too many virus babies get in the inside? Burst. Right. It's going to boom because it's going to blow up. Okay? And when it does that, what happens to all the virus babies? No. They're spread, right? Okay, so you would think they would die because they have no host cell, but that's what's going to happen next. When they explode, the host cell explodes, and then the viruses go everywhere. They go find another host cell because they can't survive without one. That's why.
It's going to cause the cell membrane of the host to rupture, explode, lyse. And so what happens is now these new viral copies, they get out of the cell and they go and infect other cells. This is very bad. This is known as the lytic cycle. By the way, the lysing of the cell membrane is a very bad thing for a cell. Cell cannot survive without its cell membrane. So definitely not a good thing for a cell. Now, the other cycle is called the lysogenic cycle. And I like to remember this is the longer word. So I like to think of this as the longer, sneaky cycle. A lot of times when we're talking about this cycle, we're talking about a special kind of virus.
Okay, so sorry, it's not pausing for me. Alright, so we got lytic, which was short and sweet, explode, boom, bad. Okay? Lysogenic, longer word, longer, sneaky cycle, and that's where the viruses hide out in your cells for a long time. Okay, that's what they're going to explain in a second. Lysogenic, I just want to make sure you got the two words down. Lytic and lysogenic. Good here? Yeah. Okay. I know you guys are listening and writing and doing a really good job with
It's called a bacteriophage. It sounds very fancy, but what it means, it's a virus that goes after bacteria. In many cases, when you're talking about a lysogenic virus, this is what you're talking about. But these types of viruses that go through this cycle, they do kind of the same thing at the beginning. They inject their genetic material, but this time the genetic material stays hidden in the host DNA. And so what happens is when the host replicates, it also replicates the viral DNA. And eventually what can happen is it can be triggered to go into the lytic cycle, and then the virus can start being assembled, and then it can lyse out of the cell. But in the meantime, whenever it's during the lysogenic cycle, it's just hidden in there. It's kind of a sneaky type of thing. So as far as what triggers it to go into the lytic cycle, it can be a variety of things, and scientists are still doing a lot of studies with this. Sometimes we've noticed there are environmental triggers like UV radiation or chemical triggers, but it can cause now suddenly this virus to switch into the lytic cycle and come out. So going back to the cold, which again is typically caused by a virus, a lot of times it has to run its course and your immune system will take care of it. But some viruses are extremely complicated, like HIV, the virus we had mentioned earlier that can lead to AIDS. That virus, its hosts that it goes after are immune cells. And
Not really a fun fact, but this is a fact to know. HIV attacks T cells. HIV attacks T cells. If you're not writing that down, write that down now. I'm out of room. I got a little bit more room on the board here. So HIV attacks T cells. And T cells are immune cells. We're going to talk about a little bit more next week. So helper T cells are what they're called. They're helpful to your body. They actually do a really good job of making antibodies to bad things that are in the environment and to protect you and all your other cells. All right, so HIV is going to kill the thing that's like the most helpful cell in our body. That's why it can eventually lead to AIDS, which is like almost a total immune shutdown.
by going after immune cells, it makes you very vulnerable to other infections and diseases. In fact, someone who had HIV, the common cold could be a very detrimental thing to their health. So it's something that scientists really are researching to try to treat. And it's difficult too, because HIV is a virus that tends to mutate quickly. And so if the medications are very selective against one specific type of virus and the virus mutates, it makes it difficult to treat. So with our talk today about viruses, you might wonder, well, is there anything positive about viruses? Well,
Yes.
Viruses do play a major role right now in gene therapy. It's a very big field right now that's really growing with research. So viruses do play a big role with that. Also, scientists are looking for ways to manipulate or engineer viruses. For example, there's a type of virus that goes after insects. It's called the nuclear polyhedrosis virus. Highly encourage you to look it up. Well, this virus is very selective. It goes after certain insects. But if it can be targeted to certain types of insects, the types, say, that eat your crops, the pest, then it can make a great form of a pesticide. Now, the only problem is whenever you add some kind of pesticide, whether chemical or viral, there can be major consequences in the ecosystem. And when we get to ecology, we will talk about that. So just something to think about. But viruses are a major component of scientific research right now. That's it for the amoeba system.
Alrighty. Okay, so you should have had like a million facts on this stuff. The viruses have they have a lot of interesting facts. So we're gonna talk about some of those. We're gonna do this worksheet in just a second. You guys wrote down capsid. Thumbs up.
Yes.
Capsid is a protein coat. It's like a little protective coat. So you need to know it's protecting the virus. You wrote down that it's not living. Good. Did you write down? Okay, so this is not this red part. That's fine. Did you draw yourself anything? Draw yourself. This is called the bacteriophage. Okay. Draw yourself the bacteriophage or phage. There's actually full college courses on what a bacteriophage is and how it can be manipulated and what are the details of it. And as you are drawing it, label the facts of the structures on it. Actually, how about you can keep drawing it? I have this worksheet. I totally forgot that this structure is on here. So if you want to draw it down, that's fine. If not, you're going to get a worksheet with it and you're going to label it. Okay? So as you go through, pass these down for me. I'm going to hit the highlight from the worksheet as we go. You can draw it, but you're going to see the labeling of it here in just a second. There is a good picture here. When you get the worksheet, you'll see bacteriophage.
Oh boy.
Evil. All right, on your worksheet right now, look. What is A pointing to on the worksheet? Chandler. What's it pointing to on the worksheet?
The capsid.
Okay, what is B pointing to on the worksheet, everybody? The nucleic acid, right? It's either DNA or RNA, and you don't know. Actually, you kind of can tell in that picture what is it? Is it RNA or DNA? Look in the picture in your worksheet.
DNA for a double helix.
Right, DNA says it's a double helix. Good. Part C, and you can write on this worksheet. This is yours, okay? Yours for the keeping. So write all over it, color it, whatever. Okay. You can even cut this out and put it in your book if you want to. Okay. So A is the capsid, B is the DNA, C.
Tail fiber.
Tail fibers. Good. Tail fibers. You got the three parts. This is A. This is B. And this is C. All right, and then in your images, your image here, let's fill in the blanks. Let's do this together. Remember that these are different kinds of viruses and virus structures can vary. Fill out the following sentences regarding labels A through Z of a common virus shown here. Label A is the what? Capsid, and its function is to what? Protect, right. Protect the DNA. Good. So B is the DNA, and its function is to, what does DNA do? I heard code. Code for, okay, I'm going to say code for genes or proteins or replication or whatever. It can be anything. It's going to code for the information you want the virus to be able to do. C is pointing to the tail fiber. Based on what you observed in the video clip, tail fiber is important to do what? Right, attach to host cell. Good job. All right, now on the right hand side of the page, you have the little pictures that you saw in the video. Let's order these in the way that they actually sequence, so they actually happen. So look at that for a minute, look at all of them, and you guys can tell me what 1 through 4 is. All right, is it the first quadrant, second quadrant, third, or fourth? That's the first step. First step, good. This is the first step because you have to attach first. Where's the next step? The fourth. The fourth quadrant is the next one because it's not been, you don't see any any viruses in there. Yes, this is the second one. Labeling this on your paper, yes? Okay. Where's the third step? The third quadrant. And the fourth one? Why? How did you know that was the fourth one? Right, it lice. It burst open. Okay. So I think the pictures are pretty self-explanatory, but I'm going to say inject here. Yes.
I have a question.
Yes.
After it injects the DNA and stuff, what happens if something takes
To what?
the virus after the phage?
The phage itself, would die. It would probably disassemble the pieces of it. Like the protein coat would fall apart, and the proteins on the legs would fall apart, and then it would be used to regenerate other viruses later on. Yeah, it would probably just dissipate. Okay, so 2, what is happening?
It's injecting the
All right, sorry, I put inject over here. This one's, what do you guys want to say? Thank you. Attach, inject, and you're injecting your genetic material here. Then what is number 3 doing? Replicating, making more virus babies. Replicating virus. And then 4, lice or burst. Good job, good words. And then those virus babies are going to go do what?
Same thing the first one.
Right, go and attach and inject elsewhere. Good. The fourth part, number 7, compare and contrast lysogenic with lytic. All right, so what do you guys want to say is a big difference between lysogenic and lytic? Right. Hold on. One of them, both of them can involve bacteria. Actually, they didn't say that, but hold off on that one. What were you gonna say?
lysogenic is a much longer process.
Right.
It can be a much longer process than lytic.
Right. This one's gonna be a longer period of time. Lytic is gonna be shorter. What were you gonna say?
The lytigenic burst itself faster.
Okay, so this one's going to burst fast, which is related to the short time period. Longer here. So what is the virus doing while it's just waiting around? What do they say? It's replicating with the host, right? Whatever, it's just going to stay inside the host body, and when the host replicates, it replicates. But it's not going to replicate rapidly because then it would burst the cell. So it kind of just hangs out, stays hidden. It does not burst the cell. What else did I say? Oh, this one, lysogenic, it usually has a trigger that sets it off to be burst, okay? This one's going to burst, lytic's going to burst no matter what. But this one's going to have a trigger that can cause it to kind of activate and burst. What were some of the things that they said it could be triggers? UV, environmental things like pollutants. I'll tell you one thing that a lot of people say. You guys know that the cold sores they're herpes, okay? The cold sore is a herpes virus. It's not like sexually transmitted herpes. There's like 3 different herpes viruses, okay? So herpes is actually a lysogenic infection. People get the little cold sores every now and then. They don't always have it. You know, they don't always have that virus just doing its thing, okay? The virus hides, and then when that person is exposed to usually some other when that person is exposed to some other factors or a lot of stress, they get a cold sore. Who's ever experienced that? Okay, so it's different factors in your life can cause that trigger, and then the virus comes out of hiding. That's all it is. So an example would be a cold sore. Okay, and if you've never had a cold sore, you will never get a cold sore unless you interact with somebody who has an active cold sore. Okay, so someone has an active cold sore, don't use their chapstick. Don't kiss them. Don't drink after them. Don't eat off a fork or something. But if they have an inactive cold sore, you should be okay. I've never had a cold sore. Cross my fingers, knock on wood. I will never get one because I don't want to touch anybody who has one at the time. Yeah. Question?
Is it like you have it multiple times, or is it like it happens once and then you don't get it?
Cold sores, they will usually be reoccurring. It's very rare to have it one time, but what happens is there has to be that trigger to set it off. So if you don't experience a trigger, maybe you only get it one time in your life, which would be awesome. Okay? Yeah.
Cold weather be
Cold weather can be a trigger, right? Environmental changes. Okay, you guys take a minute. There's one part we didn't do. You guys are smart. Figure out the prokaryotes and eukaryote one. Take a minute and do that. You have the upper quadrant you still have to do. No, keeping it in your book. And then we have one more thing we're going to write about viruses. 1 second.
what about number 8?
Oh, did I not do number 8? No. Oh, I'm sorry. 8. HIV is a type of retrovirus, meaning retro has RNA in it. Good. So you could put just RNA. It can cause an individual, stop beating the desk. It can cause an individual to become vulnerable to secondary infections. After watching the video clip, why can this occur?
Because the virus attacks the T cells.
Yes, the virus affects the T cells, so their immunity is totally suppressed. So what you would write is attacks T cells are in the immune system. And it said that the virus mutates quickly. You remember that?
Yeah.
So if it mutates quickly and we come up with a vaccine, last thing you have to write about viruses, we have vaccines of viruses. If we come up with a vaccine and then the virus changes the way it looks, the vaccine, will it affect it? No. So we come up with a nice vaccine for, let's say the flu. You get the flu vaccine. Then next year, do you have to get the flu vaccine again? Yeah, you do, because the flu has mutated over a year. That's why every year, every other year, most people get the flu vaccine. That's because we have to retrain the body on what the flu looks like at that time, because the flu is going to change and change and change and change and change. So, while you guys finish that, I'm going to pass out I have an optional extra credit project if you would like to participate. That is up to you. An example of it is here. I have Salmonella typhi. What it is, it's called the microbiography. You have a microbe that you describe and the disease that it causes. It's detailed. For you guys, it'll be due on Wednesday the 10th. I hope Tuesday the 10th. It's the 10th, which is next Wednesday. If you would like to participate, you can let me know. If not, it's extra credit, no skin off your back, but it's a choice. And at this point in time, we're going to be the end of grades, and I want to make sure that you have some options for yourself. Okay, so you're going to keep these worksheets and this thing I'm talking about now. If you have your crossword, you can leave it on the desk for me. We're leaving in about 5 minutes. We still have time here. Wait. We still have time. All right, does everybody have a worksheet? No. I ran around and checked. I marked out the only bit. No, you're kidding. All right, guys, I'm going to wait on you. Sometimes you don't transition well, so I'm going to wait on you. All right, everybody has you see the microbiography poster project. You have that in your hand, right?
Yeah.
I'm going to explain both of these sides, okay? The bacteria review and PROTIS, we're going to do that slowly. What I would recommend is making plans for the weekend to start that. I'm not going to take it up on Monday because Monday I'm taking up chapter 19 vocab, which is on PROTIS, okay? So chapter 19, that's the topic. Chapter 19 vocab is on PROTIS and fungi, and that's something that we're going to talk about on Monday, and we're going to do some labs with that, so I need you to have some background. But this review we will eventually get all the way through, so if you have some time, I would work on it. If you're packing up, you're being rude, please sit down. You're going to hold up the whole class, unfortunately. Okay, So I'm going to explain this project if you would like to do it. So what you do is, I have this on my website already, so of course don't try to type in the links. You can go click on the link on my website. You have to choose a virus or a bacteria or something that causes illness, for example, MRSA. And then you have to go through and to give me some details on the disease, the infection, what are symptoms, what are treatments, how can I prevent it? And you have to give me details on the microbe, 4 characteristics, what's the scientific name, give me a picture, what are some things it can do, and then the product. Guys, being rude, wait on me. Thank you. The product can be a poster or it can be a 3D image like I have here. This is HIV, this is Yersinia pestis, which causes a bubonic plague. Last year somebody made a little, this is the chicken pox, made a book out of it. It was pretty cute, so I was really excited about that one. So a product means some sort of physical thing I can hold, okay? It can be a poster, something like that. Now, that's all up to you. It's on your own time. If we have some time in class next week, Monday or Tuesday, I would totally let you work on it. You can research on the computers. But if you have questions, I would make sure you ask me on Monday, okay? So, I'm going to show you a couple of images of bacteria. This is due on Wednesday the 10th, if you would like to participate. I'll go ahead and pack up now. I'm going to show you some images while we finish up. Oh, it's an extra credit project, so it's going to be like a whole new another test grade. It's pretty significant. Yes. Pack up and then sit down so I can show you my little I Love Bacteria website so you can get some ideas. I have 1 minute and I want to show you some of these. ILoveBacteria.com is the best. You will find so many ideas. So let's click on Meet the Microbes. Let's look at some of these. Let's look at, okay, guys, you're standing up and I'm going to wait on you. Sit down, please, and thank you. All right, turn around. Look up here. I want to see your eyes. I'll hold you after class. I don't really care. Wait. This is something that you want to look at, okay? On the side, it says good guys and bad guys, and then you guys can choose a couple. So, for example, the Black Death, there's some cute little images of this. Jermaine, see me after class. Bye, guys.
What about the progress reports? What about the progress reports?
I said leave it on your desk.
I gotta be somewhere.
Where are you going?
Well, I got lunch, but I gotta go.
So you can look for that on Monday. Bye.
Miss Emma,
Yes.
is there a request to be moved up? Because I'm having a really hard time seeing with that.
Yeah. Let me see what I can do. Let me write myself a note. Where's my little sticky note? Let me
I
write
noticed
myself a
that
note.
yesterday, too.
Is it like you can't see because of people's heads or
No,
because
it's-
of the distance?
It's more because of the distance.
Okay. Gotcha.
So,
Have a great weekend.
Bye, you too. Where? You just give me a
I
spot?
don't really care.
Bye.
Somewhere near the front, I guess.
Like, would second row be okay?
Yeah.
Okay.
Just so I'm
I'm gonna say first or second row, and I'll move back.
All right.
Thanks, Cole.
Have a good weekend.
You too.